My name is Shreya Jain, and I'm from the Aquatic Omics Lab at Ontario Tech University in Oshawa, Canada. Today I'm presenting my master's thesis project, Refining Adverse Outcome Pathways using Japanese Medaka embryos exposed to 2,3,7-A tetrachlorodibenzoadioxin, also called TCDD. Industrial-scale chemical production is a global concern. Chemicals can act as potential hazards to human health and the environment. Dioxins, PCBs, and PAHs are known to cause adverse effects such as cardiovascular issues, cancer, and ultimately mortality. Although pathways of toxicity remain unclear, they're often conserved across species. A great way to capture the effects caused by these chemicals are a framework known as adverse outcome pathways. These pathways offer a solution to prevent data duplication and allow for grouping of data based on species and similar compounds. Adverse outcome pathways promote collaboration as it is open source and used by scientists in academia, government, and industry. And therefore, this framework may be useful in the future to prevent adverse effects in the environment caused by chemicals. An AOP in itself captures an adverse outcome caused by a variety of chemicals on a multi-organismal level, from the molecular through to the ecosystem level. A molecular initiating event, or an MIE, is what commences the inevitable domino effect, which leads to the adverse outcome, or the AO. In between are the key events, shown here in green, which are often weakly linked. An MIE that is repeatedly found in the AOP framework is the activation of the Aryl Hydrocarbon Receptor, or the AHR. The Aryl Hydrocarbon Receptor, or AHR, is a transcription factor which is found in the cytoplasm as a complex. It responds to many changes in the body, including circadian rhythm, oxygen tension, and redox potential. Ligands of the AHR are found naturally through the diet, by free radical formation, and through enzymatic activities. Chemical pollutants, such as dioxins, PCBs, and PAHs, may act as ligands as well, where binding can result in adverse outcomes. Contradicting pathways, or pathways that may be working in conjunction to a common AO, are often seen in the AOP framework. Activation of the aryl hydrocarbon is seen in a variety of organisms, such as fish, mice, and chicken. The AO observed is mortality through altered cardiovascular development. I sought to investigate this MIE of AHR activation and this AO of mortality through altered CV development or cardiovascular development by investigating two AOPs. Where AOP21 suggests altered cardiovascular or CV development through COX-2 induction, AOP150 suggests reduced ARNT-HIF1 alpha dimerization and further downstream effects as culprit, both ultimately leading to mortality. I wanted to refine these AOPs using the model Japanese rice fish. They're wonderful organisms that provide one-cell stage eggs daily, which hatch in 9 days. Their entire genome is sequenced, and they're housed at Ontario Tech University. Notably, their heart development is complete in just 7 days post-fertilization, or 7 DPF. We hypothesized that a short exposure may provide detection of the smallest differences between the two AOPs. Morula is an early embryonic stage where the cells have just divided into a solid mass of cells. This was the stage chosen for exposure, and embryos were exposed to a range of dioxin for 1 hour. 2 days post-fertilization and 7 days post-fertilization were chosen as observation points. 2, as the embryo has just gained a heartbeat and begun onset of blood circulation, and 7, as the heart development is complete, including the pericardial sac. After the exposure, embryos were washed 3 times in clean media and kept in incubation until collection for GPCR or Proteomics on 2 days post-fertilization or 7 days post-fertilization, or until hatch for malformation and hatch time analysis. Heart rate video was also recorded on 7 days post-fertilization. Some interesting findings. Here we have a graph of hatch time with hatch time on the y-axis and the TCDD concentrations on the x-axis. The concentrations tested were 0.001, 0.01, 0.1, 1, and 10 ppb. And here we can see that there was a significant delay in hatch time, which is significantly noted for 1 ppb and 10 ppb. The p-values are noted here as 0.03 and 0.003 respectively. Here we have a healthy fry up top with a healthy oil droplet, spinal cord, yolk sac, and heart. Below, we have a fry that was exposed to 10 ppb TCDD for 1 hour at the 4-hour post-fertilization mark. Tube heart was often observed more frequently and more severely in the 10 ppb treatment, as seen here. Other malformations also visually seen were the curvature of the spinal cord, craniofacial malformations, and yolk sac edema. Qualitatively, pericardial edema was seen in all concentrations. An interesting observation visually observed that I noted was hatched from the highest concentrations of dioxin tended to be missing the healthy oil droplet. This may indicate altered lipid metabolism due to TCDD exposure. More frequently, more severe pericardial edema was seen in the higher concentrations, especially in the 10 ppb treatment group. The chi-square test provided a p-value of 0.01, while a non-parametric test showed that the median pericardial edema score had increased from 1 to 2 in the 10 ppb group from the control, with the p-value being 0.01. Heart rate data was collected using an open-source heartbeat software and showed a significant decrease in heart rate from the control across all concentrations of TCDD tested, with 3 of the 5 groups having a p-value of 0.001 or less. Although the proteomics results are preliminary, many proteins in the 2 days post-fertilization samples are involved in triglyceride mobilization and chylomicron remnant clearance. Volcano plot results of control versus 10 ppb showed 3 proteins had been significantly downregulated with an FDR-corrected p-value of less than 0.05. These are shown here in the graphs. ANXA1 plays an important role in innate immune and inflammatory responses, while CRAS regulates cell growth and differentiation. OTOG plays an important role in auditory and vestibular functions. These preliminary results provide some insight, however, more analysis of the data is still underway. 3 target genes were tested in qPCR: AHR, HIF1 alpha, and COX2. qPCR shows significant differences at 10 ppb. Increased activity was seen for AHR at both stages, with significance particularly at 10 ppb, with p-value of 0.1 for 2 days post-fertilization and less than 0.0001 for 7 days post-fertilization. No activity was seen for both stages in HIF1 alpha gene. Increased activity was seen for the COX2 gene, but only for the 10 ppb 7 days post-fertilization group. This had a p-value of 0.002. Following the AOP's bottom-up, hatching time indicates RAO and is significantly delayed at the 1 ppb and the 10 ppb treatment groups. Moving up, altered cardiovascular development was observed by decreased heart rate seen across all treatment groups, and also by pericardial edema scoring, which was significantly severe in the 10 ppb group. COX2 induction was seen for the 7 days post-fertilization and not for the 2 days post-fertilization samples in the 10 ppb treatment group, while HIF1 alpha activity did not change across all treatment groups. Finally, AHR activity increased in a dose-dependent manner across treatments for both stages. All in all, these results indicate AOP21 appears to be followed as opposed to AOP150 in this experiment. Refining these AOPs will help us in improving our ability to respond to chemical contaminants more effectively to prevent adverse outcomes in humans and their environment. Further research avenues include investigating lipidomics, stages in between, optimizing proteomics, and investigating more genes by targeted qPCR. I want to thank all these individuals for helping me set foot in the field of research, and especially Professor Danina Simmons for having me in her lab. And thank you everyone for listening.